About Bezisterim effects on biological age, excuse me, Alzheimer's epigenetics, and neurologic assessments. It's hard to believe it's been 24 years since I started working on this program, but in 2000 we asked, why does dehydroepiandrosterone show anti-inflammatory, insulin sensitizing, neuroprotective, and anti-aging effects in mice, and yet not in humans? And Harvard Medical School published a review of all of the human DHEA trials and found none of these activities. I was working with Raymond Daines at University of Utah, who was funded by NIA, and he was trying to figure out how to develop longevity based on what was happening with mice with DHEA. So we discovered that the problem was due to differential metabolism leading to the neurosteroid, which is beta AET. It's not working. There. Which is the main metabolite in rodents, but we are much more interested in sex steroids as humans, and so the metabolism for humans goes down to testosterone and estradiol, and we're not making this compound that has all of the activity that they thought came from DHEA itself. Human adults have picograms per mil of beta AET, and this decreases with age. If you administer beta AET to humans, and you have to do it by injection, it doesn't have good oral bioavailability, it decreases serum cholesterol in healthy subjects. So we thought, hey, we're off to the races with a lipid drug. Unfortunately, when we went to dyslipidemic subjects, there was no activity. So this had to be another pharmacology problem, and we realized that due to chronic low-grade inflammation that we've heard a lot about today, you had an increase in 17-beta-hydroxysteroid dehydrogenase, oxidation of the C17, and that inactivates the compound, and so you lost all your activity in dyslipidemic subjects. So we went to medicinal chemistry, and we synthesized the 17-alpha ethinyl derivative, which we call bezisterim, and that endowed improved metabolic stability, oral bioavailability, and blood-brain barrier permeability. We know that our molecule binds to extracellular regulated kinase ERK1 and 2, MAP kinase 3 and 1, just to confuse everybody. And the MAP kinase, mitogen activated protein kinase activation of nuclear factor kappa B, leading to tumor necrosis factor production, is a pathway associated with diseases of aging. Bezisterim is known to decrease inflammatory signal transduction through this pathway, but it does not inhibit homeostatic activities of ERK, NF-kappa B, or TNF. Importantly, for what I'm going to tell you, bezisterim activity has been demonstrated in monocytes and macrophage lineage cells. And this pathway is also seminal for obesity-induced insulin resistance and type 2 diabetes, as well as neurodegenerative diseases. Because bezisterim does not inhibit the homeostatic effects of this pathway, it has a well-tolerated safety profile to date. Bezisterim has been shown in clinical studies to improve motor activity in Parkinson's disease and to provide positive signals in Alzheimer's clinical studies. Today, I will focus on exploratory data for the epigenetic effects of bezisterim in dementia. In a 14-week open-label study in mild cognitive impairment and Alzheimer's disease, bezisterim improved neurological assessments, neuroimaging, plasma tumor necrosis factor, precuneus glutathione by MR spectroscopy, and CSF phospho-tau biomarkers. In addition, The Epigenetic Clock Development Foundation data showed a 3.4-year decrease from baseline in biological age using the Horvath skin blood clock, and also altered monocyte methylation. Analysis performed by our collaborators at the Hurdle Group indicated batch effects in this analysis, and so that confounded the epigenetic interpretation, and we had to wait for the next study to understand that. We developed additional information on neurological and biomarker measures in a 30-week study in 50 Alzheimer's disease subjects, and we were able to obtain DNA methylation data for 33 of those subjects at study completion. Analysis of 5 biological aging clocks. Here we have the skin blood clock, the pheno-age clock, the Grimm age clock, Hannum, and from the Hurdle Group, the inflammaging clock. Showed a lower biological age than placebo at completion. And when corrected for chronological age, so if you look at the age acceleration, placebo subjects showed age acceleration, but Bezisterim subjects showed age deceleration. And I want to thank my collaborators at Hurdle for this analysis and all the help with the DNA methylation analysis that they've provided for us, including their analysis of their new inflammaging clock. Bezisterim subjects had a lower biological age with clocks that are associated with metabolic and inflammatory harbingers of diseases of aging. We analyzed association of blood cellular clocks. On the y-axis, we have the clock data with clinical hematology results on the x-axis, and we found a difference in the monocyte methylome between placebo and Bezisterim subjects. The epigenetic lymphocyte and granulocyte clocks correlated with clinical hematology measurements for both treatments, but for monocytes, only the placebo subjects had a significant correlation with clinical hematology results. Bezisterim clinical hematology results for monocytes were not significantly different from baseline, indicating a difference in monocyte DNA methylation. The lack of correlation between clinical hematology and the monocyte clock in Bezisterim subject monocytes was statistically significant. And this data was consistent with a previous study and our hypothesis presented at AAIC last year that Bezisterim might be driving an inflammatory M1 to a restorative M2 transition in myeloid cells. And we'll talk more about that. We looked at DNA methylation here, and we found increased promoter DNA methylation has been associated with decreased gene expression. That's kind of almost taken for granted now, but we do not have expression data from this study, and it's awaiting our next study. Blood monocytes are less than 10% of the total leukocytes. So if the Bezisterim differential methylation is predominantly in monocytes, then the differences in gene expression may be much larger than that calculated for the total peripheral blood cells. We found that Bezisterim differences from placebo promoter methylation were almost exclusively increased. We analyzed DNA methylation beta values for promoter regions so that we could make a better idea about expression for Bezisterim and placebo subjects. We identified significantly increased methylation of 4,900 genes that have identified genes in Bezisterim compared to placebo subject. A little lazy, and it took a long time to do it, so we only curated the top 50 of those, and we identified associations with disease progression in those genes that had increased methylation and purportedly decreased expression. There were only 21 genes with significantly decreased promoter methylation in Bezisterim compared to placebo. And 6 of these genes were associated with metabolic and inflammatory homeostasis, so something that would be positive for Alzheimer's. Genes with increased promoter methylation in Bezisterim compared to placebo Alzheimer's subjects were significantly enriched in the Kyoto Encyclopedia of Genes and Genomes, that's abbreviated KEG, and we'll speak more about that, Alzheimer's disease pathways. So these are pathways for metabolism and diseases based on proteins. The average increase in beta value for the 1,317 CpGs involved in this was 5.6%. So we're seeing kind of a small change in a large number of genes rather than a big change just in one gene, which is kind of the normal therapeutic concept. There was no significant enrichment for genes with promoter methylation that was decreased in the Bezisterim compared to placebo AD subjects. The activities of the key genes in the KEG-AD pathway with higher promoter methylation in Bezisterim included genes affecting amyloid beta, apoptosis, neurofilament light chain, and neuroinflammation. The mitogen-activated protein kinase activation of nuclear factor kappa B leading to tumor necrosis factor production is a common pathway for diseases of aging. Bezisterim is known to decrease signal transduction through this pathway. This slide shows the beta value increases along these pathways. And so what you can see is there are small changes in sequential phosphorylations in signal transduction rather than just knocking out one of those. So you have compound sequential decreases in expression of the signal, the MAP4 kinase, which phosphorylates the MAP3 kinase, which phosphorylates the MAP2 kinase, which phosphorylates MAP kinase, which activates transcription factors, which then transcribe cytokines and chemokines. And so that sequential compounding must have a major effect on these inflammatory pathways that we believe occur predominantly in macrophages, microglia, and astrocytes, leading to neurodegeneration. Importantly, analysis of a number of unrelated transcription factors, ones that are not associated with this inflammatory cascade, had less than 1% increase in beta values. So it's not a general effect to increase methylation of all the transcription factors. There's some selectivity here. It may be beneficial, in fact, to have small decreases in multiple genes in a pathway rather than a major change just in one target. And this actually may be associated with a favorable Bezisterim safety profile. We know that if you had complete inhibition of ERK, NF-kappa B, or tumor necrosis factor, you would have associated toxicities. So looking at the genes that were enriched out of these 4,900 in different KEG pathways, we see that we have enrichment, it would be Bezisterim compared to placebo, in pathways associated with neurodegeneration. And I'll just point out, there is a pathway for multiple diseases for neurodegeneration, and we enriched there. Alzheimer's was the winner here. Alzheimer's, ALS, Parkinson's, Huntington's disease, so neurodegenerative diseases, and also processes involved in neurodegeneration, senescence, apoptosis, proteasome activation, and TNF and NF-kappa B signaling. These data suggest that Bezisterim should improve diseases of aging, particularly those associated with neurodegeneration. So what did we see clinically? These are graphs of our primary and secondary endpoints in this study. I have to tell you up front that we didn't have enough subjects. It was not powered to obtain statistical significance. But Bezisterim subjects showed improvement, so they're in blue, compared to placebo, which are in red, over time in neurological assessments, including the global statistical test, the clinical dementia rating sum of boxes, the Alzheimer's composite score, the ADAS-Cog 12, the ADCS activities of daily living, the ADCS Clinician's Global Impression of Change, and the Neuropsychiatric Index. So all of these changes, we saw an improvement. Some of the scales actually go the wrong way, so we've adjusted them so improvement is always up, because it's a little bit hard to follow otherwise. Sorry, I did want to make one more point. Although the per protocol population was not powered to obtain statistical significance, the magnitude of these changes are comparable to those of recently approved Alzheimer's disease medications. In the per protocol subjects from our 30-week study, exploratory analysis demonstrated improvements from baseline in metabolic inflammatory markers, including fasting glucose, cholesterol, homeostatic model assessment for pancreatic beta cell function, and HOMA2 insulin sensitivity, and for inflammatory monocyte chemoattractant protein 1. These slides not only will be available to you at some point after this meeting, but they'll also be on the BioVie website within a day or so. So you have a little bit longer to take a look at them. We performed exploratory analysis of correlations between changes in the baseline biomarkers that are listed here and neurological assessments. And we did that separately for bezisterim and placebo. And so these are the changes that we saw in placebo, highlighted in yellow here, significant in placebo and not significant in bezisterim. So the changes in placebo but not bezisterim correlated with neurological assessment decline, and the examples given are glial fibrillary acidic protein, phospho-tau217, and C-reactive protein, which correlated, and increases in those markers correlated with decline in placebos. Conversely, biomarker changes from baseline in bezisterim, but not placebo subjects, correlated with neurologic assessment improvements. And the examples here are decreased neurofibrillary light chain, correlating with improved neurological assessments, and decreased cholesterol, correlated with improved mini mental state exam. The chemokine RANTES has both pro and anti-inflammatory CNS activities, and there is a developing literature that gives evidence that depending on the environment, RANTES can alter the astrocyte-driven microglial M1 to M2 transition in the brain, and papers in Alzheimer's as well. And interestingly, for placebo subjects, the increase from baseline RANTES was associated with decline in the clinical dementia rating, but for bezisterim subjects, it was increase in RANTES was associated with improvement, and not only in CDR sum of boxes, but the other neurological assessments as well. There were no placebo clinical changes with a skin blood clot difference in chronological and biological age, 0. But for bezisterim, the decrease in the skin blood clot differential between chronological and biological age correlated with neurologic improvements. These data suggest that bezisterim might be altering promoter methylation of biological aging genes in concert with improvements in cognition and function. We identified 18 bezisterim clinical measure change from baseline correlations with individual CPGs from this study, and with genes that were beneficial, with beneficial associations for Alzheimer's disease. 3 of those have to do with what looks like a pathway to turn on GLP-1, interestingly. We also identified 6 placebo clinical measure change from baseline correlations that were associated with metabolic, inflammatory, and dementia biomarkers that may be detrimental for AD. So we saw what we think might be bad things in placebo and what we think may be good things in Alzheimer's, in bezisterim. The data suggests that bezisterim might be increasing promoter methylation of genes in concert with improvements in cognition and function. So in summary, bezisterim may recapitulate in humans beneficial effects of DHEA that have been seen in rodents. It is known to decrease inflammatory signal transduction and restore insulin sensitivity. Bezisterim was well tolerated. We have not identified any SAEs related to the drug in a few hundred patients to date. And it's shown a biologic age deceleration compared to placebo in Alzheimer's. It may act through RANTES to stimulate an M1 to M2 transition in monocytes and microglia. Bezisterim subjects have higher promoter DNA methylation of genes involved in neurodegenerative pathways and improved correlations of neurologic assessments with inflammatory and metabolic disease and biologic aging. Bezisterim may improve Parkinson's and Alzheimer's clinical measures, but additional powered confirmational studies will be required to have any confidence in that. In general, Bezisterim may improve what we call systems dysregulation that we think is a hallmark of Alzheimer's disease to restore homeostasis through expression changes of key neurodevelopmental gene products. And this may be the first study to demonstrate evidence of improvement in longevity and clinical benefit in the disease of aging. I want to stop at this point and thank you and my collaborators. And because I am over, maybe we'll wait for questions until the break and see if we have time for that.
Thank you very much for a very interesting talk. And yeah, maybe we would have time for a very quick question. Otherwise, we
I hope that doesn't mean nobody understood anything of what I said.
Otherwise, there's a question there in the back.
I'm sorry, I can't hear you.
Maybe, can you take the microphone, please?
Did you use the original Horvath skin and blood clock, or did you use the principal component version for this testing?
No, this was the original skin blood clock. It's calculated by the Epigenetic Clock Foundation in the reports back to us.
Thank you.
We have done principal component analysis and have a very interesting story, but I could talk to you later about that.